Genome India Project, a very interesting topic, a very important topic, right? So from the topic name itself, it's very clear that it's a project in India, right? And related to the genome. Now, the most important question which will arise in the mind is that what is a genome first of all, right? Why government is going for this particular project, isn't it? And what are the benefits of this particular project? Right, so for that, let's have a look at what this particular project is. So in this particular project, the Secretary of the Department of Biotechnology has said that Genome India Project has done more than 7,000 genomes have been sequenced, right? So in this particular project, Indian government is going for the sequence of 10,000 genomes from across the country, pan India. People will be selected anonymously from around the country, and their genomes will be sequenced. The questions are, what is a genome, what is genome sequencing, right? All these questions are definitely there. So now, first of all, let us understand what is a genome. So genome, as it's written over here, is a complete set of genetic material or DNA. What is this complete set of genetic material or DNA? So genome is made up of its individual components, genes. Now what the question is, what is a gene? So first of all, let us start from the basics. So this is the diagram of a DNA. What is the DNA? DNA is the basic unit of genetic information which is over there, right? It's made up of, what are the four components it is made up of? So it is made up of, one, the phosphate molecule, which is, and the sugar molecule, phosphate molecule and the sugar molecule, right? These two form the basis of DNA, which is written over here, as you can see, right? It's formed of the sugar molecule and the phosphate group, right? And the third important component of the DNA is the nitrogenous bases that we have over here, right? So there are four nitrogenous bases, which is, one is adenine, which is represented by A, then cytosine, guanine and thymine. All right, these are the four nitrogenous bases with respect to the DNA that we have. Now let's have a look at the DNA. So a very important and a very specific point over here is that these nitrogenous bases have a very specific pairing. All right, so adenine, it's called ATGC, as you can see over here. A is going for T only. Over here also A is going for T only. Over here also A is getting attached to T only, right? So ATGC is the pair. A will only attach with T and G will only attach with C. All right, so adenine is going to attach with thymine and guanine is going to attach with cytosine. And this is the entire sequence. So DNA, as we know, is a double helix structure, right? So it has one strand over here as it is going like this, and then we have the another strand, right, which is going like this. In both the strands we have these nitrogenous bases. These strands are made up of phosphate and the sugar, and on these strands we have the nitrogenous bases. All right, so these strands, nitrogenous bases, and then this nitrogenous base, in this particular pairing, they're attached to the two strands, right? So the question is, what is a gene now? So one particular sequence in this particular DNA, let's say from here to here, all right, this particular portion can become a gene. So gene is a sequence. To understand it very easily, let us compare it with the help of a book. So let's say we have a complete book. So book will be the genome. The entire book is going to be the genome, right? DNA, let's say, is going to be one page in that. All right, one page in that. And gene is going to be one sentence in that particular page. So in this manner, we can very easily understand what is a gene, what is a DNA, what is a genome. So DNA is this particular structure. In this, various sequences act as gene, right? And when we have the complete information of all the genes, they will act as genome, right? So now let's have a look at it over here. So what is the gene? What is the importance of gene? They help in encoding proteins, and proteins are the building blocks of the cell. right? They're very important for all the key functional molecules, our biological processes, the way we look, the color of our eyes, the color of our skin, how our body functions, various organ functions, all this is decided by the genetic material, by the proteins that the gene is encoding, right? So that is the fundamental of how we function at all, right? So that is the gene, and the entire information of all those genes is going to be genome. Now, one very important point over here is that not all genes are going to be encoding for protein. So there are two types of gene. One type of gene will be encoding for the protein. Basically, it will give directions as to what kind of protein has to be created. And there are other kind of gene which are known as non-encoding genes, right? They will not encode for proteins. Then what are they going to do? They are basically going to help other genes in encoding for their protein, right? They can also help, in the structuring of the DNA, how the DNA is being packed. So they have various other functions as well, not just encoding for the protein. So all these genes, coding, encoding protein, whatever genes are there, even some genes would be non-functional. Non-functional in the sense, they do, whenever we do research, we might not find that, what kind of functions do they really have. So some may be non-functional, right? So even that can be there. So all these genes combined together will form what is known as genome. That is why it's known as complete set of genetic material, or the DNA that we have, right? So now comes the question that what exactly is genome sequencing, right? So when we are able to understand that this particular gene is going to code for this protein, this particular gene is going to code for this protein. So when we have this entire information of what kind of gene is going to code for what kind of protein, that entire thing is known as gene sequencing. And when we do this for all the genes we have, that will be known as genome sequencing. All right. So that's all we have understood. Now let us have a look at what is this Genome India project. We have understood the basics. Let's dive into the project. So this particular project is inspired by the Human Genome Project. Yes, already a project of this scale, even larger scale, has already taken place. This was there, started in 1990s, right? And it has already ended. So in that, they took this sample from across the world and went for gene sequencing of almost all the genes which are there in the human body. Guess the number of genes which are there in the human body. So it's more than 20,000. More than 20,000 genes are there, functional genes. Now this I'm talking about the functional genes, okay, which are encoding for the proteins, right? So more than 20,000 functional genes are there in the human body, or entirely available, right? So this particular project, the Human Genome Project, has already done this particular process. But why then the Indian government is going for Genome India Project? Because the Indian population would of course be less represented when we have a project of the world scale, right? So only few people from India would have represented that particular project. So basically India wants that more and more representation of its own population. It wants to understand the genetic diversity or genetic variation in its own population, right? And that is why India is going for Genome India Project. So it will collect the genomic data of more than of over 10,000 people from across the country, right? And they will identify the genetic variations and related health outcomes. What are these health outcomes? So since gene is responsible for the entire functioning of the human body, they will be responsible for the various diseases which are taking place, right? They will be responsible for what kind of effect the drugs or the medicines that we take are going to have on the human body. For all these genes will be responsible. So if we can understand the genetic variation in the human body, can we not understand the the impact drugs are going to have? Can we not make personalized medicines? All these things can be done by understanding the genetic variation in the human population, in the Indian population, and therefore we can make drugs, personalized drugs, which are suited for the Indian population, right? It will also help in understanding the genetic diversity and the evolutionary history of the Indian population. So how have we evolved, how the genes have changed throughout history, all these data can be known from this particular project, right? The question is, how are we going to do it? Let's not get into the details of it. Let's not get into the technicalities of it, right? We can only understand the name. which is like, let's say, DNA sequencing, genotyping, phenotyping, data collection. All these things are going to be there. These are the various methods through which we do the genetic sequencing, right? So that's all for this particular project, this particular topic. It was very important, few very basic concepts, very important concepts were involved, and I think this is going to be very important from the exams perspective.